Thank you, Garrett. Okay, yeah. So before we get into our panelists, I just want to give a brief overview about research, just very briefly, especially for those of you who are freshmen or just haven't had a research experience before. So just some things to think about. Why research? Why do so many pre-med undergrads participate in research? Well, it's a really great opportunity to sort of express your curiosity, inquiry abilities, just ability to be a scientist. And that's something that a lot of medical schools like to see. So it's just a really cool way to sort of explore those aspects of yourself and, just grow your base of knowledge. And who can participate? Anybody can participate. UT, as you'll hear about probably sometime during this presentation, has even a freshman research initiative, but there is a place for everybody within research. You just got to find, the things that you might be qualified for and do a little bit of digging if it doesn't seem so right off of the bat. But anybody can participate in research. And how can you do research? Well, you can do research here at UT. You can do research at other universities, hospitals in the area, like clinical jobs as well. But there's a lot of different ways that you can get involved. And again, our panelists will talk a lot more about that. But it's a lot, again, just like doing research on your own to find out what labs are available or like have openings, using online databases and resources to see what programs are out there. So it's kind of just taking initiative on that. And then when can you do research? You can do research throughout the year, like all of our panelists do as well. And there's also a lot of summer research programs specifically for undergraduates that you can participate in too. So that's just a very, very brief overview about research as an undergrad. And then if you want to, these are just some links that you can access after this presentation that are linked on the AMSA research guide, which is the first link on the slide. But these are just some resources out there for you to, if you haven't already, start looking into different programs on and off campus. Eureka is like the main research database for UT research opportunities. And so you can go in there and really tailor your interest and see what's out there. It's not, I don't think it's updated regularly. At least when I used it, there were a lot of things that kind of didn't seem up to date. But so don't let that be like a definite example of like what is out there. But that just is one resource that you can use. And then there's also the Office of Undergraduate Studies. They do information sessions, and they're definitely willing to help you get involved with research if it is your first time, as well as they do offer like a lot of different research scholarships and stuff. And then there is the Freshman Research Initiative and the Accelerated Research Initiative, which I know will be touched upon later, but those are just some more structured research programs for undergraduates here on campus. And then the last two links are just some databases for summer undergraduate research programs across the country at different universities. And so you can definitely look into those if you want to get involved with a summer research project. Those are pretty cool too. So I see the chat has a couple of comments. Cool. Thanks, Garrett. Yeah, so if there's not any immediate questions other than what was in the chat, I will go ahead and hand it over to our panelists. So I'm sure all of you guys know these people already, but if you would like to, each of you, just like for a brief moment, turn off your mics and just introduce yourself. I don't know, say like your year, major, which is already up there, and then just a fun fact, and then we'll jump into the individual slides.
Yeah, I'm Garrett. I'm a junior. I'm a biomedical engineering major. And a fun fact about me is I really like playing like .io games. It's pretty like relaxing, like Augur.io, Slither.io. So I do that a lot.
All right, hey everyone, my name is Andy. I'm a junior mathematics major, but I'm still pre-med. Actually, yeah, so kind of weird, but cool. And a fun fact about me is that I've recently figured out that when I crack my back this way, it cracks more than when I crack it this way, which is really weird. And I think something might be up with my spine, but I'll figure it out.
That sounds interesting. My name is Ro. I'm a junior. I'm a neuroscience major. I guess a fun fact about me is I'm also a classic civilizations minor. So I have a special interest in that, and that has led me to learn how to write and read hieroglyphics.
Pretty cool. My name is Krishna, and I'm a junior biochemistry major. Fun fact about me is I love riding horses, and I do so every summer.
And Senna, and then
I can just jump in. Sorry,
Okay.
I'm like multitasking. Okay, hi, I'm Senna. I'm a sophomore neuroscience major. I always have a hard time with these fun fact things, so mine's not very fun, but I'm Turkish, so that's pretty cool.
I think that's really fun.
Thank you.
Thanks, guys, for introducing yourselves. And we'll go ahead and just jump right into the first presenter, which is Garrett, and I'll hand it over to you.
Yeah, sorry if camera quality is pretty bad. It's just dark, I guess. I can't really fix it. But yeah, so to jump right in, I started out wanting to do research because I thought it'd just be a cool thing to try out. I heard a lot of people trying it out and thought it was interesting. Biomedical engineering has a lot to do with research. It's a really research heavy major. And so I thought I'd just give it a try. I really wanted to do something with coding. I had always been interested, but I never really got a whole lot of practice or practical application with it. Only took a few classes, but I really wanted to make it a part of something I do on a daily basis. And I also wanted to connect it with medicine in some way. And so what I did was I researched and looked around the biomedical engineering website at different labs that they have there, specifically in the computational engineering side of things. And I emailed around a few labs. Some labs, emailed me back. Some, I don't know what happened. I did find one lab, and the professor was super nice and willing to have me jump on, not on any of his projects, but he did know someone who did need help with projects, and so that's who I work with right now. They just asked my email. They just asked for my resume and looked at it, and were like, "Okay, yeah, it's fine." And so I jumped into the UT's computational oncology lab, which kind of works with MRIs, radiology, and uses those MRIs for to predict cancer treatments and model cancer growth or disease in patients, and try to quantify it and create some quantitative models to, in a sense, help treat cancer. And so what I specifically work on is breast cancer. I take breast cancer MRIs and make predictive models about different treatment outcomes. And so the patients that I work with, they go through neoadjuvant therapy, which is NAT. And I basically look at their MRI scans from each of their 4 visits to the hospital where they get chemo treatment or neoadjuvant therapy, which is a mix of different treatments. And so the way I do that is through getting a huge data set of MRIs and looking at them, and then also looking at them. And the way I look at them is through MATLAB, and I take those images and make some predictions. So on the right-hand side, this isn't necessarily from one of my data sets, but it's similar to something that I might do, where they are looking at a contrast agent put into the breast cancer or into the breast tissue. And so the contrast agent makes things brighten up a lot better so you can see it. You can see the tumor is actually getting a lot smaller through each cycle of neoadjuvant therapy. And so you can model that in different patients to see if the tumor will go completely away or still have a little bit of tumor left. And if you can predict that, you can change their treatment to something that might be more helpful or stronger, or not as strong if you can predict what the final outcome might be based on like the first 2 MRI scans. And so it's really interesting stuff. Typically what my week looks like is usually I spend around 8 to 10 hours a week doing research. I still do research because it's mainly computational, so I just email my professors, send him PowerPoints. I work alone on this project, so it gets a little lonely, but I do have a few contacts that I can talk to, grad students, my professor, who all are very helpful if I get stuck on anything or if I just want a second opinion. And so, but it is nice to have kind of my own independence, and I can really like take the project where I want it to go and push it. And so typically I spend, 8 to 10 hours coding, looking at different research papers in the general area of radiology and computational oncology, and direct contrast enhancement, which is just to do with kind of what I'm doing. And so I do that mainly just to get a good feel of what other people are writing and putting out there, and other experiments that other people are doing to hopefully give me some ideas about what to do with my own work. And so I really love it. And so my tip, and so that's really helped me to keep going with research. And so my tip for y'all is to find something that you're actually passionate about. And so, a lot of people will kind of just get into research to check a box off the to-do list for like pre-med or getting into grad school. But I would advise not doing that. I mean, it can work if you're, a box checker and you really want to just do it just to put it on a resume, that's fine. But I highly suggest you might be more productive and be able to get a lot more done and enjoy it better if you actually care about it. So I would definitely research around and actually do something you want to do. Second thing, your PI and grad students are there to help you to learn and to grow. And so I know I've gotten a lot of mentorship from my research PI. I think he's a really cool guy. And just how he works has really been inspiring to me. So reach out to them and tell them what you're interested in and what you want. And so yeah, and then also just work hard. Research does take time, and what you put in is what you get out. I try to dedicate 8 to 10 weeks, but people, that's just my story. That's not everybody. Some people do more, some people do less. But work hard and show that you care, because that can go a long way to getting like more projects and more assignments and building that goodwill with your professor. Additionally, something that really helped me, a little thing, is just to find papers to read. If you're going in and you have to interview for a particular research lab, it might be helpful to look at their website and look at the papers that they've written, so that you can kind of keep the conversation going about like what they might do, like what they're doing in the research lab and what you might be able to do and help with. And also finding papers to read just helps you be a better researcher. In research, you have to do a lot of writing typically, because if you can't communicate results, then it's kind of pointless. So for me, that's really helped with my writing and just communicating results in different graphs. So yeah, I'm pretty sure that's all I had to say about my research. But I would definitely recommend getting involved, or at least trying it. So yeah.
Thank you, Garrett. I think Ro has a question. I don't know.
Yeah.
I did not see it.
Yeah, no. I just raised it. I raised my hand. But so what exactly does this predictive models kind of look like coding wise?
Yeah, so it's kind of, it was hard for me to understand the coding behind it at first. I didn't really know a whole lot of MATLAB, but one thing we look at is relative intensity of MRI like brightness, I guess. So MRI images have different voxel or pixels that have different, or we call them voxels if they're 3D images because it's volume. But so pixels have different values associated with them, and so brighter ones typically have higher values in our MRI scans. And so what we can do is look at, okay, so a contrast agent is put into the tissue, and then you take an MRI scan over a period of time. And so that's a temporal scan. You look at the changes in the breast cancer, in the tissues. And if like one area gets brighter than another, you can kind of say, oh, there's a lot of vasculature going on there. And that means that the tumor is dangerous and growing and being, has blood vessels going to it, which is not good. And so, but, on the other hand, if it's getting, the values of enhancement are getting smaller, then things will, then things are looking good for the patient. And so we look at that, those values at each little pixel. And so computationally, that's really expensive. And so that's it's a good thing we have MATLAB to do that for us.
Thank you.
Are there any other questions for Garrett? That was awesome breakdown of your project, Garrett. That was really cool.
Thanks, Ron. Thanks.
Clap, clap, clap. Okay, so we'll move on to the next presenter, which is Ro.
Oh, cool. Okay, so hi, my name is Ro. I worked in Dr. Aldrich's lab. I'll start with where I first got my research launched from. I worked in, for a summer, I worked at MD Anderson. We worked with secondary acute myeloid leukemia, excuse me. And we were focusing on targeting BET proteins and transcription factors in order to essentially stop growth, excessive growth, and induce apoptosis of cancer cells. And so if anybody has any questions over that, I have 2 abstracts that were published for the American Society of Hematology, I believe, ASH. So feel free to ask me about that later if you wish. But for what I did at the Aldrich lab is Dr. Aldrich focuses on ion channels. And specifically, his lab focuses on the structure of ion channels. And we understand, what the purpose of an ion channel is and how important it is for signal transduction and for creating your action potentials. And, it's very, very important, but we don't know exactly the nuanced details of their structure. Many ion channels, specifically the calmodulin and calcium-based ion channels, they have these different subunits that seem to be very similar, but perhaps they may actually have different purposes. And that is kind of what this lab is focusing on trying to uncover and, focusing on uncovering and seeing if that there is any sort of like alteration and difference in each of these subunits. So what I've got over here is one of the things I was doing was a personal kind of a fishing project, which is basically just kind of like a project that kind of sees if there's any leads to take that we can kind of further expand onto a project. My mentor asked me to do this. And what you're seeing is this is a very small cap, but what you're seeing is loss of function. Yes, you don't have to do wet lab or biological scientific research. Yes. So what you're seeing here is a bunch of loss of function mutations in the coding of one specific KCN1, I believe, or it's, and I believe that stands for the small potassium ion channel. It is a family of 4, and this one is the first of the family. And what you're seeing is loss of function mutations in healthy, phenotypically healthy individuals. So there's this database that essentially did a genetic sequencing of a bunch of phenotypically healthy individuals. And since you're seeing a loss of function mutation, this shouldn't result in a healthy individual. And what this means is that perhaps these certain areas in the genetic coding could be pliable. And so what we do is we like to mess with the terminals. We like to make NN terminals, CC terminals, and we like to mess around with these proteins and see what that does, and if that, how that changes it can lend us information as to what the function of these proteins and these specific subunits do. So this was kind of an independent kind of data analysis. So in the duties that I did, so analysis from a genome sequencing database, and other things I did was protein purification. I did PAGE gels, which is polyacrylamide gel electrophoresis. I did photospectroscopy of purified proteins, and then generally maintaining lab equipment and keeping everything clean and other sort of like small errand duties. So with the most common type of lab performance you're expected to do is just the general obligations of what you consider a lab technician, who basically just kind of run things and just do things for other higher ups. So it's a lot of just bench work. But honestly, I'm one of those people who absolutely love bench work. So that was amazing for me. So my tips into getting into a lab is just to listen out for professors mentioning colleagues or mentioning their own work. If you have a professor that mentions their research, ask them about it a little bit, and then kind of maybe go to office hours and ask them, hey, you mentioned your research, do you have any openings? If you're their student, they're more likely to kind of let you in, or at least tell you about it, and maybe they'll keep in touch with you and let you know if they do have options open. How I got into Dr. Aldrich's lab is because he mentioned that his colleague was open to positions. And so I jumped on that, and I want to piggyback and just not piggyback, but rather just confirm what Garrett said about doing a little bit of research ahead of time into papers that the PI is published, because getting into a conversation with Dr. Aldrich, I think it very much helped knowing what his research was about, knowing kind of where he was going, what things that he was interested in, and kind of just discussing it with him and showing him that, I do have an interest in his lab. Because the last thing the last thing PIs want is to bring somebody in who just wants to check off a few hours and will leave. They kind of want somebody who's going to be there for a little bit longer. So, adding and kind of showing that you're committed and you're, willing to stay for a good amount of time to learn and keep helping them, because it is immensely helpful if you know what you're already doing and you know how the lab runs. It's just going to make things easier for them instead of having to keep training undergraduates. So kind of try being in it for the long term. Try, doing a little bit of research ahead of time. Reach out to as many people as possible. It's very much a throw shit at the wall and see what sticks kind of situation. So just reach out to as many professors and check their lab websites, because often, like Dr. Aldrich's lab website literally had we want undergraduates, please apply and ask us. So, go to these lab websites and you can see them. I think if you go to a department and go to their faculty and see research, and you can look up Dr. whatever lab and just look at their websites and see if they say maybe there is a application process or something of the sort. Just try to look into as much of the websites and information that's already online before you start reaching out to them. And that might help streamline the process. But yeah, and this is just the SK or KCNN1, or rather just an SK channel. And then this is just further, just kind of looking at the ion channels that we tend to deal with. It's we deal with a lot, and basically anything calmodulin based or calcium based. But it is fun work. I absolutely adore it, and if you can get into research, I just highly encourage it. It teaches you so much. I think that's the end of that. Sorry, I might have talked very fast.
No, that was wonderful. Thank you so much, Ro. Does anyone have any immediate questions? Okay. So we'll move on to our next presenter, which is Chris.
All right, thank you.
Krishna?
I think we lost you, Carissa.
Oh.
Yeah.
Hello?
Oh, yeah, you're back.
Hello?
Yeah, you're back.
Oh, okay. Thanks for letting me know.
Okay, I'll just go ahead and get started. All right. Hey, guys. My name is Krishna. So over the past 2 semesters, I've been conducting research under the ARI program, which is the Accelerated Research Initiative. And this is pretty similar to the FRI program, except it's catered to people who haven't taken FRI. So if you haven't taken FRI, this is a great place to get some research in. The stream that I selected was practical sensors. And what we did, we tried to create an indicator displacement assay approach that can measure the concentration of an analyte in a solution using UV-vis spectrophotometry. It's kind of a mouthful, but pretty much what it means is we're trying to detect the concentration of some substance inside the solution. And our goal is to try to find the concentration of tartaric acid and malic acid in wine. And one thing that I learned is that for research, it's applicable when people can make money off of your product. That's when it really sells. So this is great because traditionally, if you wanted to find a concentration of a solution, of a substance inside of the solution, you can use some, you can use NMR or like high performance liquid chromatography, HPLC, stuff that require heavy machinery. However, the problem is that it requires heavy machinery and you can't have it field ready. Our goal is to create an assay that can work for field ready approaches where you can do it out in the vineyard and on the grapes and find the best time to harvest the grapes. So if I can direct your attention to the images on the right side, the second image from the bottom represents an indicator displacement assay. So what you have is in dark gray, you have the receptor, and that's bound to an indicator that's in light gray. Now, in the presence of some sort of analyte, which is in this case tartaric acid, the tartaric acid will compete for that receptor spot and displace the indicator. Now, when this indicator is displaced, it actually changes the color of the solution, and that can be detected by a spectrophotometer, which I've kind of illustrated a brief, an overview for what a spectrophotometer does on the top. You can see that if the light source shines through a solution that has your analyte inside of it, you can use it to detect the concentration of absorbance. And this is shown in the graph at the very bottom. Now, you can see that as the concentration of the solution or the substance inside of the solution increases, the absorbance is also increased. And the whole goal of our experiment was to create this host receptor complex. And I loved it because it didn't just have me doing one thing continuously over week after week after week. First, you have to develop, try to manufacture a host receptor complex, and then you have to use next week you come in, you test it with different solutions, you test it with different indicators and see if it actually works. A lot of the times we failed, but at the very end, we finally created a good host indicator complex. So that's thumbs up. But pretty much my experience, what I've learned is that the mentor is expected to be full of questions. And this is really important because I really didn't know anything about indicator displacement. I didn't know what indicator displacement assay was. I used spectrophotometers briefly in some labs. However, not to this extent. The great part is that the mentors are there to help you because in turn you are helping them. So just ask as many questions as your heart desires. They're there to help you. Also, like Garrett said, you want to find something that will interest you. So specifically, this was really great for me because you're always doing something different week after week after week, and it's to one set goal. Unlike some general lab classes that you guys will be taking where you kind of just do one experiment per week, write a lab report, and then move on to a different experiment. This used different techniques, but it's all for one big goal. So I love that part of it. And you want to find something that kind of tailors to you in a way. This was interesting to me, but in the practical sensor stream, there's also different sub-branches where this was the Borch project. There was also this NASA project where you can actually use practical sensors to detect life signatures on different planets. And that was really interesting. Some people might like that. There's also a branch where you can create peptides, which is very useful for grad research. So all in all, there's a lot of different options, and I think ARI gives you a great, great selection to choose from. Also, you have very flexible schedule, so it's great because specifically for my lab, you can go in pretty much whenever you want and conduct the research. You just have to talk to your mentors. It works around your schedule because at the end of the day, you're helping them, so they're pretty lenient. The benefits of doing research specifically in ARI is that you get to learn how to use different lab equipments, different lab tools, which you can use in further research later on. Additionally, you gain experience researching on your own, which is really important because oftentimes you come across a question that you and your mentors don't know the answer to, and you have to just reason possible solutions and test it out. And I think that's a great, it involves a scientific method, and it's great because you get to apply that in a physical concept. Another great part of ARI is that you meet other streams and other researchers, and it's a great way to network because when you get into this program, you automatically are in access to different streams, different people who potentially you could research with later on if you talk to them, if you're interested in their work. Just ask them some questions and they'll be more than happy to take you on, especially since you're already doing research with someone else. And a big part of this is obviously the course credit. It's great because specifically this stream gave me credit for OChem lab, which I know every pre-med has to take. And I also got credit for AChem, which currently I'm taking. And if you're a biochem major, it's a great way to knock those two out. Also, later on, you can open for paid mentorship experience as well. So after you do this lab and you've learned all the tools necessary, the mentors love to get you on board and then train the next people who are coming up. And all in all, that's the ARI program, man. Hope you guys like it.
Thank you, Chris. I see an internet connection unstable thing again. So I don't know if people can hear me or not.
Good right now.
You can hear you.
Okay. Okay. Well, okay, cool. Thanks so much, Krishna. Does anyone have any questions? Okay, so move on to our fourth presenter, Andy.
Awesome. Hey, everyone. So my name is Andy, and I do a lot of work in bioinformatics. That's the lab that I'm under. We're technically under the Department of Integrated Biology, which is a fancy way of saying we use computers to do biology research. And so specifically, I'm in the Hoffman lab. It is the Hoffman, the same Hoffman that you hear about when you take biochem. I don't work with him directly, but I work under him. So that's kind of cool. But I specifically work under this person called Dr. Young, and she's amazing. She used to be part of the FRI program. So she's super cool about helping out undergraduates. And I'd recommend contacting her if you're looking for resources because she's really cool. But a bit about my research. So what I do is proteomics research, which is a fancy way of saying using data to look at proteins. And so the specific project I'm working on this semester is a comparative analysis between sexes. So essentially, we're looking at chicken eggs and trying to figure out what the protein mechanism is that determines the sex of the eggs. Because they found out that in some species of birds, the eggs come out by gender in a specific order, where you'd have sometimes it be the case where the first egg is always male or the third egg is always female. And that was really weird because you want to know why, like if sex is usually like a 50-50 chance, why does it still come out in a specific order? So it's kind of strange. But there are a lot of biological underpinnings, like the environment could be a factor, sort of the order is definitely a factor, but why that is a factor is still unknown. And so what my project is looking at is trying to figure out what proteins are involved and sort of where these pathways are and what they are. And I'm working specifically with chicken eggs because they're super cheap. And what essentially we did was we took out the proteins from the egg yolk and we ran them through a mass spectrometer, which is a fancy machine that just tells me what proteins are in there. I didn't do it, of course, because I'm working remotely, but well, my PI did essentially, so that's cool. And we also know what sexes the eggs are. So essentially, we're going to divide the eggs proteins up by sex and figure out what the differences in those proteins are. So think about like protein abundance. Do male eggs have more proteins in this area more than female eggs, or vice versa? And so a lot of it is heavily computational because there's no way you could do this by hand. And what essentially I do is I go to this program called R, which is a statistical programming language. I put in the data and I do a bunch of math that I don't understand just yet, and it'll tell me things about these proteins. And on the right, you can see one of these figures that I've made. It's a network heat map plot, which is a fancy way of saying I've taken these proteins, I've made a tree diagram, which essentially orders the proteins based on their relation to each other. And then I've made this new plot, which is a heat map that characterizes how related proteins are. So the darker colors mean that the proteins are more closely related, and the lighter colors mean that they're less closely related. And this is mostly for aesthetic purposes. You can't really tell anything from this, but it looks really cool. Most of the actual results are numbers, but I figured this would be more interesting to show. And so you can see these pretty colors where this clustering software algorithm that I used has put specific sort of groups of proteins in specific clusters. And what we're really hoping to find is that there are clusters that are responsible for the different sex differentiation in the eggs, which essentially just means we're looking for a cluster that contains the proteins that determines the sex. And hopefully we can figure out what those proteins are and then do actual like knockout tests or something like that later on with real biological samples. But for now, this is mostly sort of a exploratory project. This is really fun for me because unlike some other projects that I've been in, this one I really had to start from scratch. Like this is something my mentor started this semester. So I've really been on since the entire beginning. And I've been working like from the raw data all the way building up to this finished, well, almost finished product that I have right here. And so it's been a pretty long journey of 4 weeks, 4+ weeks, whenever August was at this point. And it's been a great experience. What I do also want to point out is that this isn't my first lab that I've been in. So I was part of the FRI program years ago when I was a freshman. So I know a little bit about that. And I've also worked in the anthropology department under the primatology department specifically. So I've also done a little bit of work in that. So if anyone's an anthropology major, maybe you'd be interested. But yeah, what I want to really emphasize though is sort of the stuff down here. So it's never too late to start research. I mean, I know I started like in the FRI program, so I'm hardly like a good example, but I didn't really find what I was passionate about until like this year in my junior year, right? I was in FRI because they had course credit, and then I was in the other lab because I was just trying to get any research position. But like this semester, I really made like a conscious choice about what subject I wanted to research. And I think that's really important because you will be in here for at least a semester, whoever you choose to work under. And so you want to make sure that's a good decision. But don't be afraid to jump around labs, right? So I have been in a couple of labs, and I haven't been in a lab at this point longer than a semester, which might sound bad, but isn't really that bad. I mean, you can go like one pathway where you find a lab you're really interested in, and that's what you want to do, and you can stay like 4 years or whatever, and that's super cool. But you can also keep looking around to find out what you're really interested in. Because, for example, I'm a math major, so I like biology, but I also like math. So trying to find things that weren't just bench work all the time, no offense, Ro, was one of my interests. So I've been kind of hopping around, and yeah, and like that's totally fine. I've talked to a bunch of grad students, and what's really important in your undergraduate years is to figure out what you like. Like that's more important than staying in a lab you don't like for like 3 years straight, right? It's better to figure out now before you actually commit when you go out to like grad school or something. So don't be super chill about that. Also, yeah, so to answer Carissa's question, I'll be staying in this lab the entire year, but I heard that the COVID modeling consortium has openings, and like as a math major, that calls to me. So I'm not really sure yet. I'm still trying to figure that stuff out, but I'll definitely be in this lab for a while because I love the mathematics that's used in bioinformatics, right? Like bioinformatics kind of sneaks up on you because you know it's computer based, but there's a lot more math than I bargained for, which is great for me, but maybe for people who are more biologically inclined, that might not be so great. It's kind of like when you take ecology and like you figure out there's like stats in ecology and you're like, what? But yeah, same kind of deal, lots of math. But I'm not gonna belabor the point, so I'll just close out right here. Yeah, definitely talk to grad students. They're like super wise and they're super chill, and they'll tell you anything you want to know about like what being a researcher is. And last but not least, there's this program under the TIDES department or TIDES organization on campus called TEJAS, and it's a work study program where you can get paid to be a research assistant. So like if you want to not have a separate job but get paid to do research, that's something you want to look into. The program's quite small, and like I didn't they basically have like 100% acceptance rate because not a lot of people know about it. So definitely something to look into. So yeah, I'm gonna close out with that. Any questions? I did not. What was the question? Once I'm a sophomore, you can switch between streams. It's not super common. If you've been in it since a freshman, that's something you'd want to talk to your PI about, but it is possible. Yeah, for FRI.
Thanks so much, Andy. Nobody has any other questions. We will hop over to our last presenter, Senna.
Hey y'all. So my name's Senna. Like I said, I'm a sophomore neuroscience major. I'm kind of the baby of this panel, so hopefully I can give y'all some good insight. This is my first lab that I'm a part of, but it's a little different from everyone else's labs. My research isn't as like scientific and like super smart sounding as everyone else's, but I think it's still pretty cool. So I'm currently a member of the Clínica España de Epilepsia Pediátrica. I work under Dr. Karen Shea, which is a super dope board certified child neurologist at Dell Children's. Similar to Andy, we're starting up right now and we aren't an established lab, but we're working on it. We expect to be open on December 1st to everyone else in the greater Austin area. So the whole purpose of my lab is to understand healthcare disparities that exist for families of children with pediatric epilepsy, specifically Spanish-speaking families, because living in Texas, there is a big Spanish-speaking population. So as a healthcare provider, you definitely want to be able to cater to that population and make sure that any doctor's visits they have are super smooth and they can comprehend everything that's going on, especially with such a hands-on type of illness like pediatric epilepsy. So that's what we're looking at. Surprisingly, there have been very few studies that have specifically looked at these health disparities with pediatric epilepsy. There have been a lot of adult epilepsy studies, like among non-Hispanic Black or Hispanic adults, but very few when it comes to pediatric. And there have been no pediatric studies directly assessing or reporting rates of pediatric epilepsy, specialty referrals, or visits by race or ethnicity. So there's not a lot of like research about what we're doing, which I think is pretty cool. So we're not like building on anyone else's research. It's a bunch of like cool, fresh new stuff. So all that to say, my role in the research is I speak to all the families prior to, during their visit. We are aiming to have about 400 families involved. We have 4 like trial groups of about 100 individuals. So I ask them a bunch of questions and document their responses so we can report it into REDCap, which is the research program that we use. And I'm also responsible for following up with them every 3 months for 1 to 2 years, and we expect them in the clinic every 6 months or so. So on the screen, you can see it's the little like survey that we fill out. So the left side's in English, the right side's in Spanish, depending on what language the family speaks. And if they're a Spanish speaking family, the entire experience is in Spanish for them, or if they have like a translator to do it with them. So it's like a lot involved, but we try to make it a very like immersive experience for the families. So some of the things that we've done, being just a little bit of a startup, is our PI was able to get a little over a $79,000 grant from Dell Children's in order to set up this clinic. We have 5 different clinic areas around Austin and Round Rock and Cedar Park, I think. And then 2 of my fellow researchers created scales, which are kind of like the scripts that we follow in both English and Spanish. And there's at least 30. They're so long and hard to get through. And then we're also overseeing the possibility of converting into a multi-center trial involving a hospital up in Chicago. So getting to see this process unravel and see it start to form on a greater scale has been pretty cool. We've had to discuss a lot of incentives for participation and figuring out how we're going to incentivize these families to participate, since this is a pretty long project. And our latest issue has been finding another nurse that spoke Spanish to be able to join our team, but we resolved that. We have a super awesome team of providers. So that's kind of like the few things that we've been going through, like little growing pains as we've been like kind of starting. But I think we're pretty established now, so all we need to do is get approval and we should be good to go. So this is my first research lab, but I think I've learned a lot in the process of trying to like join the lab. Oh wait, sorry, I'm like ignoring the chat. The families are expected to participate in the project for at least 1 to 2 years, because that's how long the duration of this project is supposed to be. So that's what we're hoping. But it might stretch out longer. It depends on if we kind of get the information that we need, I guess. But moving on to a few things that I've learned, a few of my tips kind of reiterate what the other panelists have said, but I think that it's really good advice and it's worth hearing a few times because I think it's super important. So the first one is a lot easier said than done, but it's don't be shy and to go after what you want. I knew that I wanted to do research when I got to college, but once I got here, I got super shy and super intimidated. I didn't want to join an org. I really wanted to join AMSA as a freshman, but it took me all of freshman year to build up the courage to apply. So that's why it took me until being a sophomore. But I feel like I've missed out on a lot of opportunities to join something meaningful just because I was shy and intimidated. So hindsight's 2020. So like after reflecting a little bit, I knew that if I wanted to do something, like I had to go seek it out, and like being shy wasn't an option. So I had to pull the extrovert out of me, but it pays off. So definitely do your best to not be shy and exhaust all of your resources. It was very overwhelming to me how many resources UT exactly had. I know Carissa touched on a few of them at the very beginning of this presentation. UT has a lot of opportunities, and so does AMSA, for everyone to succeed. So definitely seek them out. And even if you don't think you're eligible for it, look at it. And I say that because I found this research opportunity on the Bridging Disciplines program. I'm not sure if you guys are aware of what that is, but I'm not even in the Bridging Disciplines program. But I saw the post on it because it was directed to just like UT students, and I guess she uploaded it there. So yes, it is really cool. So I like emailed her and I reached out. So it's definitely worth asking, which leads me to my third point. If you're not sure, definitely ask, use those resources. And if you're not sure if you're eligible or if you like know exactly what's expected of you, to definitely ask because it wouldn't hurt. And then my next point is to make sure you pick a research group that you resonate with. So we're expected to have our clinic open for about 2 years. So that means by joining this group, I'm committing the next 2 years of my undergrad to the research project. So it definitely needs to be something that's super worth it for you. And I personally resonate with this whole project because it is something that runs in my family. My older brother had epilepsy since he was a child. So I definitely have a personal tie to the research, and it just makes it that much more I don't know, worth it whenever I'm working on it, and it keeps me wanting to do it. So make sure it's something that you completely support and you want to do and you want to go through, so you can keep putting in that passion and that you don't get burnt out with it. And then finally, I looked at so many research groups before I found this one and was fully content with this one. So it's really important to not give up and keep on trying, because UT has a ton of research projects, even outside of UT, like mine's associated with Del Med, not even UT specifically. So it's aimed toward UT students, but it doesn't mean it has to be through the university itself. There's a lot of other research opportunities that aren't specifically through the UT system. So make sure you look there too if you're having a hard time finding something for you. And just remember that being pre-med is super duper hard. I'm only a sophomore, and I know how much of a pain in the butt it is. And I'm sure everyone that's older than me can attest to that. It gets worse. So being pre-med super hard, and finding cool positions that make an impact are hard to find also because of how competitive like being this major is, like by itself. So don't ever give up, and remember that if it doesn't work out, to just keep trying, and you'll be really glad you did once you find something that you like.
Yes, that very, very well said. That was awesome. Thank you.
Thank you.
So we have about 5 minutes left, and I would just like to open the floor for everybody to ask any questions that they might have for any of our panelists about anything, about their project specifically, or about, you know, just research in general. Yeah, so please do not be shy, like Senna said. 7? I don't know if I'm pronouncing your name correctly. Please correct me if I'm wrong, but please.
It's Azine, but you were pretty close. I wanted to ask, like, just everybody in general. I know, like, you guys are all upperclassmen, so how do you balance researching and then like getting good grades and also not like dying? That's my question.
I could talk if that's cool. Yeah, so when I first started out doing research, you kind of got to ease yourself into it. I started doing like my first full-time research, like 6 hours a week in my primatology lab. And I was with a grad student who was super cool. So like whenever I had exams, she's just like, you can take the week off. Like, I got you, like you're good. And so like finding like really cool people to work with who are understanding about that is super nice. This semester, it's sort of the same situation. I'm technically on a 10-hour per week schedule, but it's like a wobbly 10-hour per week schedule where I can do less if I need to for my exams, and my professors understand. So don't worry, like if you see on those websites, you must commit 10 hours a week for the entire semester. It's not always like 10 hours a week every week forever. It's a bit more flexible than that. So yeah.
If I may add on to that, it's a matter of communication. You just have to like be a good communicator about what is going on. If you're having a particularly tough week, like it is your job to make sure that you communicate this to your PI. They understand because you're a student, you're doing student things. You're not gonna like be able to automatically commit your time. So it's just a matter of letting them know in advance. Just being like, hey, next week I have a test coming up. I need the hours that we spend, you know, in lab. I need time to just like focus on schoolwork. And they should normally. I've never met a professor that isn't okay with that. But they'll be understanding. In addition, what helps is scheduling your time out and just making sure you have for me, I had a committed schedule as to when I came in. It was like 3 days a week. I would come in for a certain amount of time here, here, and here. And it worked with my schedule. I knew those were days that it would be a bit lighter. I knew kind of what I was working with. And therefore, I created a schedule that had the least amount of conflict. And therefore, I was able to, on a, regularly commit myself to lab work instead of having it kind of be a toss up each week. And if I came in extra, great. If I was only able to do what I could do, all right. If I needed to take one of those days off, I just let people know. So Google Calendars is like really good. I like schedule like pretty much every hour of my day out. It is fantastic. So it is really great to visually see what you have going on so you don't, over commit yourself to many things. So it's just a matter of organizing your time.
Ronton has a question.
Or it's more of like a comment, because like I actually haven't done research myself, but I did get to work in a wet lab as a lab assistant, which is basically like doing all the dirty work and stuff. But I also got to like help assistant researchers with some of their lab stuff. And I think ultimately, like the thing that they're looking for is, are you putting in effort? Like, can they visibly see that you're trying to do your best? And I think that way you can like balance it, because I know definitely I would hear like all the PIs and all like the grad students and PhD students just complain about their undergrads like skipping work. And And so I think the most important is just showing that you're ready to do work and putting as much effort as you can. And that way they'll be more lenient with you, if that makes sense. But yeah, that's from an outsider perspective, kind of.
Yeah, I think that's good. I see Sothvik asked, when sending cold emails to professors, how can I work to improve my resume in order to make myself a better applicant for RA positions? Also, I'm a freshman with no research experience, also about reading up on different topics. Are there any sites or other places that I could see to find interesting things? First of all, being a freshman with no research experience is okay. I didn't get my lab stuff until I was into my sophomore year. So you have time. And a lot of these professors know as undergrads that this is going to be your first lab experience. So don't like beat yourself up over not having lab experience, thinking that they're gonna want stuff, because most people don't have never had the opportunity to get lab experience prior to university. So why would they expect that of you? As for making your resume better, it's just kind of, if you did things that kind of show you're interested in science, I'd add things onto that. But I think I think the chat has mentioned that there are templates for resumes. Those are fantastic. I think what really kind of attracts, not attracts, but like what really convinces a PI to let you join their lab is what happens in your communication. Show intense interest in your emails. Go to the interview, ask for an interview if they don't say anything. Like, just show as much initiative as you can, and prove to them that you're not doing this, and this comes with finding interest in their actual lab. So don't just do things for the sake of research, but if you like a lab, like, go for it and go hard for it.
I also want to jump in and add, don't feel pressured to do research out of the gate as a freshman. Especially since you're just coming out of high school, you only have like high school things on your resume, which is okay as a freshman. So don't feel pressure to immediately get into research or find research. Like, it takes time to find those things, and that's absolutely okay. Like, everyone's timeline is different. So yeah, just don't feel like you have to rush yourself into it. Don't feel pressured to.
I also want to add a little bit about this topic. So you don't always have to be the most qualified to get the positions, right? I know some of them might seem really competitive and some of them are, but there are a lot of people out there who are waiting for eager freshmen to come in so that they can teach them everything they know. Like, for example, I didn't know the first thing about monkeys when I was in my sophomore year applying to the anthropology department, but there are some really cool people there. And I was just saying, hey, I'm not exactly interested in monkeys, but I love like the data analysis y'all are doing for this kind of data. Like, can I join? I'm interested in that. And they're like, whoa, like, you're like math, you're interested in this data analysis, which is kind of different from what I've heard before. And you seem like you're really into this. And so, yeah, like, they took me on. And so it doesn't always have to be like, I am really interested. I'm so interested in this topic, in fact, that I majored in this topic. Please let me in. It could just be like, you're doing something really cool. I read a little bit about it. Can I be involved? And I mean, you'd be surprised. Like, some of the professors are really chill, and some of the grad students are even cooler, maybe, perhaps.
Yeah, PIs often get kind of busy. So your grad students sometimes end up being more of a mentor than PIs do, because they're busy. They often, I know at least Dr. Aldrich, he had like 3 teams working under him. So it wasn't just like one lab head working under him. He had 3 teams. So I had, usually kind of interact with like the kind of the assistant research heads, the people right under the PI, or even or graduate students. And those graduate students just got out of what you're doing, like they just got out. So it's not like they're not some sort of old professor who doesn't remember what it's like. It's very recent for them. And sometimes, they can be very great for just advice, and they're working on their own stuff. So they're not, too far away from you. So definitely talk to them, see what they're doing, ask, what you could do to get where they are.
Well, thanks so much for sticking around a couple extra minutes and for your awesome questions. I think several of our listeners have already left. But yeah, again, thank you all so much for coming, and big thank you to all the panelists for sharing. That was super informative and just a lot of great advice. So I hope you guys were able to take away some really good stuff today. But yeah, you're free to go. Thanks for coming. Have a good rest of your night.
Thank you guys. Good luck in your research.
Bye.
Very nice.